This video is to supplement your Chapter 1 reading. We will briefly introduce cell biology with this chapter. Therefore, potentially new terms or concepts will be mentioned, but not explained until later chapters this semester. We will begin with our learning objectives for this chapter. Upon completion of this chapter reading and homework, you should be able to 1. Discuss the major tenets of the cell theory. 2. List the fundamental properties of all cells. 3. Describe the major differences between prokaryotic and eukaryotic cells. And 4. Discuss the properties that distinguish between a virus and bacteria. Before we get into too much detail, I would like to acknowledge that there's quite a bit of biological diversity here on Earth. Many species thrive in special environments and have unique adaptations. This diversity is better covered in courses such as microbiology, zoology, and botany. This class will strip away any species specializations and look at the function of specific intracellular structures. Furthermore, we will focus primarily on eukaryotic cells this semester. Modern cell biology has a rich history. We will briefly survey some of the more famous contributors to our understanding of cell biology. The first is Robert Hooke. He was credited in 1665 with coining the term cell through his microscopic studies of cork. The microscope he used had a magnification of about 30x. In 1673, Van Leeuwenhoek was able to observe living cells in samples such as pond water with microscopes that he created, which had varying magnifications upwards of 300x. In 1831, Robert Brown was able to not only view individual cells in plants, but able to name the nucleus. In 1838, botanist Matthias Schleiden concluded that all plant tissues are composed of cells. Furthermore, he proposed that mature plants arise from a single cell. In 1839, anatomist and physiologist Theodore Schwann similarly concluded that animals are also composed of cells, have nuclei, and arise from an embryo like plants. We can take a brief look at some of the early microscopes used. Keep in mind the time period and as well the technology continues to improve. The first microscope shown here is one that Robert Hooke used. The image to the right is a sample of cork, which he used to observe and record his findings in his publication entitled Micrographia. The second microscope is one of several which Van Leeuwenhoek used. This particular microscope has a magnification of just under 300x. This third microscope is Robert Brown's microscope. Through the conclusions which Schleiden and Schwann made by observations of plant cells, the cell theory was proposed. It was originally only two tenets. These were that 1. All organisms are comprised of one or more cells, and 2. The cell is a basic unit of structure for all organisms. In 1855, Rudolf Virchow modified the cell theory with the statement that every cell arises from another cell. This was groundbreaking at this time because this concept dispelled the common thought of spontaneous generation. In addition to cytology, or the microscopic study of cells, two other important fields are important for our understandings of cell biology. These include biochemistry and genetics. Therefore, we will find a mixture of these fields discussed this semester. There are some basic properties common to all cells. Your textbook keeps this to a list of 9 items. However, this is more of a survey of similarities and certainly not all-inclusive. 1. Cells are complex yet organized. This is evident through the boundaries set by membranes. 2. Cells possess a mixture of nucleic acids, including DNA, which is where genes are housed, and RNA, which is important for protein synthesis. 3. Cells have the ability to divide and reproduce. 4. Cells have the ability to acquire and utilize energy. 5. Cells are capable of carrying out a variety of chemical reactions. 6. Cells can carry out mechanical or physical activities such as contraction. 7. Cells must have the ability to respond to chemical, mechanical, thermal, and electrical stimuli. 8. Cells are capable of self-regulation or maintain homeostasis to keep factors in set point range. The 9th item listed in your text is the ability to evolve. I'd like to clarify here that populations and not cells evolve. A biological definition of evolution is a change in gene frequency within a population. This is likely confusing because some populations may be composed of single-celled organisms. 10. I would also like to point out a couple more items that I think could be added to this list. One is the fact that all cells contain ribosomes to synthesize proteins. 11. The second addition is that all cells possess membranes. Once we have an idea about some common features to all cells, we can begin to look at some differences. The first is classification of cells. Cells generally fall into one of two categories, prokaryote or eukaryote. The term kary refers to a kernel or the cell nucleus. The term pro refers to before. A prokaryote then refers to before nucleus. Prokaryotic cells are those which lack a nucleus. In comparison, eu refers to true. A eukaryote has a true nucleus. Whether an organism is a prokaryote or a eukaryote, these cells will have a cell membrane, DNA, and ribosomes. Prokaryotic DNA is concentrated in an area of the cell referred to as the nucleoid region. Prokaryotes do not have membrane-bound organelles, and they exist as single-celled organisms. There are two biological domains that are prokaryotic, domain bacteria and domain archaea. In comparison, eukaryotic cells are in domain eukarya. Eukaryotic DNA is compartmentalized within a membrane-bound nucleus and have a variety of other membrane-bound organelles such as mitochondria, chloroplast, and Golgi bodies. Eukaryotic organisms may be single-celled or multicellular organisms. Domain eukarya has 4 kingdoms within it, including the animal, plant, fungi, and protista kingdoms. We'll take a quick look at the prokaryotic domains. This image is of me and my sons in front of a thermal vent in Yellowstone National Park. If you look closely, you may notice the variety of colors in the pool. This is due to the rich biodiversity of prokaryotic species thriving in the hot pool. An example of archaea living here is Sulfobulus, shown in the image to the left. Bacteria also thrive here, such as the Thermocrinus rubere. These bacteria give a pink hue to the pools. Table 1.1 summarizes the common features between prokaryotic and eukaryotic cells. Take some time to review these common features. Table 1.1 continues to point out features that are unique to eukaryotic cells. You may notice a common theme in this table that many of the eukaryotic features are considered to be more complex when compared to prokaryotes. Don't confuse complex with better. Each species has evolved over time to thrive in their specific environment. This figure illustrates a generic prokaryotic cell. You should notice that the cell is an organism. It may have an outer capsule around the plasma membrane, a cell wall, and a flagellum. Within the cell is the cytoplasm, where a variety of molecules are suspended, including the DNA within the nucleoid region, ribosomes, enzymes, and nutrients. This figure illustrates a generic eukaryotic plant cell. Plant cells have an outer cell wall made of polysaccharides, a cell membrane of lipids and proteins, and intracellular membrane-bound organelles. Eukaryotes also have a cytoplasm with a variety of molecules, ribosomes, enzymes, nutrients, and also a protein scaffold called the cytoskeleton. The DNA, however, is compartmentalized within a nucleus. This figure illustrates a generic eukaryotic animal cell. Animal cells do not have a cell wall, but do have a cell membrane made of a variety of lipids and proteins, intracellular membrane-bound organelles, including a nucleus, ribosomes, enzymes, nutrients, and a cytoskeleton. Not all cells look like the generic images just shown to you. Instead, eukaryotic cells of multicellular organisms grow into unique shapes and sizes based upon their programmed function. This process is called differentiation. In this image, a human fetus is shown. This fetus originated from a single spherical cell. Following fertilization, the single cell began a series of cell divisions. Cells grow to specific regions to form unique tissues and organs such as the placenta, brain, heart, skin, and so on. We will conclude this chapter discussion with an introduction to viruses. It is important to acknowledge them as they have a significant impact upon life. Viruses are pathogenic or disease-causing to cells. They bind to the surface of specific host cells, and viruses are structures made of protein and nucleic acids such as single-stranded DNA or RNA. The outer protein component is referred to as the caspid. Dependent upon the cycle of the virus, a virus may have a lipid membrane that it has acquired from a host cells. Viruses are not living because they do not satisfy the tenets of the cell theory. Outside of a host cell, viruses are called a virion. Less structurally complex pathogenic particles are called viroid. These are snippets of RNA, no protein or membrane component to them.